hormones produced by the pancreas. This is the pancreas. Pancreas serves two functions that are carried out by two different groups of cells: exocrine versus endocrine cells. An exocrine cell does not secrete its products into the bloodstream, while an endocrine cell does. Today, we will focus on endocrine pancreas. The endocrine pancreas consists of groups of cells known as islets of Langerhans, which are embedded in the exocrine portion of the pancreas. The normal human pancreas contains around 1 million of islets. Islets of Langerhans only make up about 1 to 2% of the pancreatic cells. Each islet contains at least 5 types of cells. Alpha cells produce glucagon. Beta cells produce insulin. Delta cells produce somatostatin. Somatostatin inhibits the release of glucagon from alpha cells and insulin from beta cells. As you may recall, hypothalamus also produces somatostatin, which inhibits release of growth hormone from anterior pituitary gland. Today, we will cover 3 topics. First, let's look at insulin. Here is the primary structure of human insulin. It consists of 2 chains. A chain has 21 amino acid residues. B chain has 30 amino acid residues. 2 chains are linked by 2 disulfide bonds. Insulin is secreted. Therefore, after transcription and translation, insulin protein go through a typical secretory pathway that involves rough ER and Golgi apparatus. Insulin is stored in vesicles before it is secreted through exocytosis. Right after translation, a single polypeptide called preproinsulin is made. In the rough ER, the signal peptide is cleaved and disulfide bonds are formed, giving rise to proinsulin. In the Golgi apparatus, the C-peptide is cleaved, giving rise to mature insulin. Here is insulin receptor. Insulin receptor is a receptor tyrosine kinase. It is comprised of 4 subunits, 2 alpha and 2 beta subunits. The alpha subunit binds insulin. Insulin receptor is broadly expressed. After insulin binds the receptor dimerizes. Dimerization of insulin receptor activates intrinsic receptor tyrosine kinase, which then phosphorylates each other. Insulin receptor functions through 3 main pathways. One is PI3 kinase, protein kinase C lambda, or protein kinase C zeta. It controls group 4 membrane localization. Second pathway is PI3 kinase, protein kinase B. It controls glycogen and lipid metabolism. 3 known enzymes are the targets of this pathway. One is glycogen synthase, hormone-sensitive lipase, and acetyl-CoA carboxylase. Third pathway is RAS, RAF, and MAP kinase pathway. One target gene is C-FOS, a transcription factor. Here, I want to point out that both PI3 kinase and GRB2 proteins contain SH2 domain. Insulin regulates metabolism of carbohydrates and lipids, as we have seen in early chapters. Insulin also promotes potassium movement into the cell by increasing the activity of sodium-potassium ATPase. Insulin promotes anabolic process and inhibits catabolic process. Here is a summary of insulin functions in the liver, muscle, and edible tissue. We have seen most of them. Here, I want to use the first three as examples. Insulin increases glucose uptake by promoting membrane localization of GLUT4. Insulin increases cellular respiration by increasing consumption of ATP through sodium-potassium ATPase, as we have seen earlier. Insulin stimulates glycogenesis by activating glycogen synthase. This slide shows how insulin controls transport of glucose. After insulin binds the insulin receptor signaling activates protein kinase C lambda zeta pathway and increases membrane localization of GLUT4 vesicles. After GLUT4 arrives on membrane, it increases uptake of glucose into muscle cells and adipocytes. After insulin receptor is inactivated, GLUT4 returns to vesicles. This slide summarizes three mechanisms by which insulin controls glycogen synthesis. As you may recall, like most enzymes, glycogen synthase is active without phosphorylation. The enzyme is inactive upon phosphorylation. Protein phosphatase 1 activates glycogen synthase by dephosphorylation. First mechanism is insulin activates glycogen synthase by activating protein phosphatase 1. Second, insulin pathway through protein kinase B phosphorylate and inactivates glycogen synthase kinase 3 or GSK3. GSK3 phosphorylate glycogen synthase. So here is a double inhibition. The end result is insulin increased glycogen synthase activity. Third, insulin pathway blocks action of glucagon. As you may recall from glycogen lecture, glucagon through protein kinase A phosphorylate glycogen synthase. Insulin activate phosphodiesterase and decrease the level of cyclic AMP and therefore decrease the activity of protein kinase A. In addition, insulin blocks secretion of glucagon. Insulin secretion is increased by amino acids and high level of glucose. Insulin secretion is inhibited by epinephrine and low levels of glucose. Now, let's move to part 2. We will look at glucagon. Glucagon is a peptide hormone. Glucagon, along with epinephrine, cortisol, and growth hormone, opposes many of the actions of insulin. Insulin and glucagon move nutrients in opposite directions. The directions of nutrient fluxes is very sensitive to insulin-glucagon ratio. In a normal post-absorptive state, this ratio is around 2. After a carbohydrate meal, this ratio rises to 10 or more. During starvation, this ratio drops to 0.5 or less. Protein or fat meal does not change this ratio significantly. Here are changes in insulin and glucagon levels in real time. Blue line represents glucose level, red line for insulin, green line for glucagon. After we take glucose, the glucagon level initially rises, but it drops quickly once the glucose level rises. At the same time, insulin level rises sharply. After blood glucose level returns to normal, the insulin level follows. Finally, glucagon level comes back to normal. Major functions of glucagon is to maintain blood glucose level by activation of hepatic glycogenolysis and gluconeogenesis. Glucagon receptor is a G-protein coupled receptor coupled to GFS. Glucagon receptor is mainly expressed in the liver and kidneys where gluconeogenesis takes place. Here is a summary of G-protein coupled receptors. Most G-protein coupled receptors described so far, including glucagon receptor, utilize GFS and cyclic KMP pathway, except these 6 receptors. Among them, 4 have been covered a few times. Here, I use ATCG to summarize them. A represents angiotensin II receptor in vascular smooth muscle. T represents thyrotropin releasing hormone receptor. C represents catecholamine alpha receptor. G represents gonadotropin releasing hormone receptor. Here is a summary of glucagon functions. Glucagon stimulates glycogenolysis by activating glycogen phosphorylase kinase. It inhibits glycogenesis by inhibition of glycogen synthase. It stimulates gluconeogenesis by increasing transcription of PBCK and G6PC genes. It stimulates lipolysis by activating hormone-sensitive lipase. And finally, glucagon stimulates ketone formation and amino acid uptake into the liver. Similar to insulin, glucagon secretion is increased by amino acid. Opposite to insulin, glucagon secretion is increased by epinephrine and low level of glucose. Glucagon secretion is inhibited by somatostatin, high level of glucose, and high level of insulin. Glucagon is one of the 4 stress hormones. When blood glucose level is low, first, hypothalamus sends a signal to pituitary gland, which then relays the signal to adrenal gland. The adrenal glands release cortisol and epinephrine. Hypothalamus also sends a signal to sympathetic nervous system, which releases norepinephrine. Second organ that senses the glucose level is pancreas, which then releases glucagon. Cortisol increases gluconeogenesis. Epinephrine and norepinephrine increase glycogenolysis. Glucagon does both. Now, let's move to part 3. We will look at clinical correlation. First condition is insulinoma. It is found almost exclusively within the pancreas. Pancreatic tumors hypersecrete insulin. It is the most common islet cell tumor. Most common symptoms, confusion, disorientation, headache, are due to hypoglycemia. Second condition is diabetes. Main signs of diabetes can be summarized in 3 P's: polydipsia, polyuria, and polyphagia. Main symptom is hyperglycemia, which causes a wide range of complications. Type 1 diabetes is caused by T-cell mediated destruction of pancreatic beta cells. It begins in childhood and progresses rapidly. Patients depend on insulin for survival. Metabolic changes in type 1 diabetes include hyperglycemia, ketoacidosis, and hypertriacylglycerolemia. Ketoacidosis results from massive mobilization of fatty acids from edible tissues, followed by hepatic ketogenesis. Hypertriacylglycerolemia results from reduced activity of lipoprotein lipase. Other changes in type 1 diabetes include hyperosmolarity, dehydration, hypovolemia, and depletion of intracellular and whole body potassium. Patients with type 1 diabetes depend on insulin. Now, human insulin can be produced by recombinant technology. Type 2 diabetes accounts for 90% of all the cases of diabetes. Major factors that contribute to type 2 diabetes are insulin resistance and inadequate insulin secretion. Type 2 diabetes is a polygenic disease. It ranges from predominantly insulin resistance with relative insulin deficiency to a predominantly secretive defects with insulin resistance. This coincides with progression of this disorder. In pre-diabetic patients, their blood glucose levels are normal. Here, this chart represents glucose level. The blue line represents glucose level from normal individuals, and the red line represents glucose level from obese people. As you can see, glucose levels from obese people are normal. Here, this chart represents insulin levels. The blue line represents insulin levels from normal individuals, and red line represents insulin level from obese people. As you can see, insulin levels in obese people are abnormally high. It is high levels of insulin in these patients that keep their glucose level under control. High insulin levels also indicate insulin resistance. Here is progression of type 2 diabetes, and this chart represents glucose level, and this one represents insulin level. If the blood glucose levels still keep rising due to worsened insulin resistance, eventually pancreas can no longer maintain the higher than normal level of insulin production. When the pancreas has exhausted its capacity, insulin production starts to fall. Then, glucose level rises even further. This eventually leads to full-blown type 2 diabetes. Diminished effects of insulin raise blood glucose and TAG levels. These patients have hyperglycemia and hypertriglyceridemia. But they still can use some glucose to partially meet their energy needs. Ketogenesis is not as robust as in type 1 diabetes. First line of treatment is to reduce caloric intake and increase physical exercise. This can help increase insulin sensitivity. Oral drugs are available for type 2 diabetes. First group of drugs promote insulin release. Second group of drugs either suppress gluconeogenesis or increase lipid storage. Third group of drugs inhibit absorption of carbohydrates. We will look at 3 drugs further. First drug is sulfonylurea. Second drug is metformin. Third drug is glucagon. Sulfonylurea blocks ADP-dependent potassium channel, which raises the membrane potential of beta cells. Depolarization opens the voltage-gated calcium channel. The rise in intracellular calcium level promotes release of insulin. Metformin decreases gluconeogenesis by suppressing genes in hepatic gluconeogenesis, including pyruvate carboxylase, PEPCK, and G6PC. G6PC codes for catalytic subunit of glucose 6-phosphatase. Metformin does so by 2 mechanisms. One is to activate AMPK. Second is to inhibit glucagon-induced rise of cyclic AMP. Glutazones activate PPAR gamma, which increase storage of fatty acids. As a result, cells depend more on glucose for energy. One downstream gene activated by glutazones is adiponectin. Adiponectin increases insulin sensitivity.